All right, everyone. Great. Let's start again. Joel, I see your slides. Brilliant. Are you ready?
Yeah, I'm ready.
That's fantastic. Okay. All right. Okay. You're coming up Apurva as well, which is great. All right. So our next speaker is going to be Joel Leal-Gutierrez from UCSD.
Thank you. I'm going to talk about genetic basis for unilateral renal agenesis and heterogeneous stock rats. URA has been described as the urinary tract malformation, and it's also coupled with some reproductive tract malformations. So it's determined as a congenital absence of one kidney. Previously, it has been reported a locus associated with this specific phenotype. It has been named as RENAG1. It only harbors the KIT gene, so this gene has been described as the most probable candidate gene for this specific phenotype. It has been also reported an insertion located in the intron 1 of this gene. It's an endogenous retrovirus. You have the representation here. It's composed by 3 different elements, 2 different long terminal repeats, and 1 endogenous retrovirus sequence. This gene encodes KIT proton co-gene receptor tyrosine kinase, which is involved in regulation of cell survival and proliferation. Introduction. Previously, Dr. Garrett was able to develop a strain. This is named the HSRA. This strain has been developed from the same genetic background as the one that we are using here, the HS population. This is important because it means that we can assume that the same QTLs responsible for URA are present in both populations. This strain was inbred for unilateral renal agenesis, and it shows an incidence between 50 and 75% of the offspring showing unilateral renal agenesis. This shows that through breeding, it's possible to increase the frequency of those elements that are responsible for URA and increase the incidence of this specific phenotype. The incidence in the present population is around 1.9% of the population. I'm going to show the ACI incidence. This is one of the 8 founded strains of the HS population. Is the one with the highest incidence. It goes between 5 and 15% of the offspring. Additionally, it has been described an incomplete penetrance for this phenotype. The objectives of this analysis were to identify genomic regions associated with URA controlling for case control imbalance, and to recognize potential loci able to modify the penetrance for this phenotype. Materials and methods, we used 5,600 individuals from the HS population. Those individuals were genotyped for 3.5 million markers, and the case control ratio that we identified was around 1 case per 51 controls. So it's not very extreme, this case control imbalance. You are already familiar with the HS population. I'm only going to talk again about the ACI strain. We used for the association test the R package and Sage GDS, applying an additive model for the association. This package is able to control case control ratio when it's extremely imbalanced, and it applies a generalized mixed model association. Here you have a comparison using the standard linear mixed model and Sage. As you can see here, the former is unable to control type 1 error. You can see an increased rate. On the other hand, Sage is able to control this error, and it's able to pinpoint highly significant QTLs associated with this type of phenotypes. This representation is showing a thyroid cancer. It's a phenotype with a very skewed imbalance. 1 case in 1,134 controls. Results, I'm showing here the most significantly associated region. It's located in chromosome 14 between the megabase 32 and 37. You have a detail here. We have again the KIT gene right in the middle. So this result is in agreement with previous reports showing that this gene as the most probable candidate gene. Here you have the most significant SNP. We had a question here since we have a total of 1,200 markers above of the association threshold. So we wanted to know if maybe we have multiple QTLs associated with URA. So we run a secondary association analysis fitting the most significant marker as a covariable in the model. And we were able to determine that the most probable result is just one QTL. This SNP was able to account for all the variability present in this specific locus. We perform a secondary analysis using haplotypic probability estimation. We focus this analysis on the associated region, chromosome 14, 32, 37 megabase. And we also focus the analysis on the haplotypic probability for the ACI strain. The goal of this analysis was to determine if maybe this specific region is more frequently inherited from ACI when we are talking about cases. So here you have a representation. This is haplotypic probability by strain. It's showing all 8 strains. I'm showing the subtraction between cases and controls. So if you have a positive value, it means that this probability is more, it's higher for cases. As you can see here, we have the ACA strain. It's more highly probable for cases inside this specific region. The second plot is showing exclusive alleles by a strain. Showing here a subset of SNPs that are ACI a subset of alleles that are ACI exclusive. Another subset is not ACI exclusive. It's present in ACI, VOF, and F520. However, when we couple this information with the prediction of haplotypic mosaic for the ACI, we are able to determine that even when those alleles are not ACI exclusive, they are more frequently inherited by this specific strain for cases. So we are able to conclude that for this specific region, the most significantly associated region, there is a higher influence of the ACI strain when we are talking about cases. As I said previously, incomplete penetrance has been also described for this phenotype. If we take a look at the distribution, phenotypic distribution for the genotypes for the most significant marker, we are able to determine that not all the cases are at least carriers for the risk allele, being the allele T. So it agrees with an incomplete penetrance for this phenotype. For this analysis, we performed a similarity analysis. The goal of this analysis is to tackle that issue of an incomplete penetrance. We used the Plum database. We fitted the Jacquard index, and we used as a reference the HSRA strain, and everything was coded in Java. So this methodology is going to tell you how similar are two sequences. So here you have a metadata example, one reference, the HSRA strain, and 2 different individuals. So you are going to perform a pairwise comparison between each individual against the reference. And the result, similarity score, it's coming from how many elements are in common between the 2 sequences that you are doing the comparison. We used the most probable polymorphisms associated with this phenotype, the insertion of the endogenous retrovirus sequence. We did this analysis in 2 different steps. We did a prediction for transcription factor binding sites in all 3 sequences using transcription factors that are expressed in kidney or kidney-related tissues such as collective tubes. For example, here we have the NO1 was discovered as one of those transcription factors able to interact with DNA sequences for those insertions. The second step was to perform the similarity score analysis. So we know that this specific transcription factor is being encoded from this specific locus, chromosome 5, between those locations. We pull out all the SNPs that are inside this locus, and we perform the similarity score analysis. So you are going to end up with a comparison for the N01 between each individual and the N01 from the HS array, the reference sequence. Here you have the results for this analysis. A number of transcription factors were identified as significantly different when you group the individuals based on the genotype for the most significant marker. They have been color-coded. The green represents the highest similarity with the HS array, and the yellow the lowest similarity. If we take a look, for example, to this transcription factor, the HNF1A, individuals that are grouped as TT for the most significant marker, they are highly similar to the HS array strain, meaning that this transcription factor had been selected for in those specific individuals. This transcription factor encodes a gene regulator of the proximal tubule, and it's involved in apoptosis. Those processes are required for organogenesis of kidney. As a result, we were able to identify some transcription factors that have been selected for in the HS array strain. The mechanisms that we believe it's related in this phenotype have been previously described in stimulating cells. Liu identified that higher expression of the KIT ligand, it can be determined as KIT activation, can drive proliferation. On the other hand, lower expression of the KIT ligand, it can drive differentiation, cellular differentiation. Those two processes are key involved in kidney organogenesis, since proliferation is required for heretic VAT invasion and differentiation is required for methanolytic rudiment differentiation. All those, a balance between those two processes are required for a normal branching morphogenesis, which is required for organogenesis of kidney. So this balance between proliferation and differentiation are key. We believe that key expression can drive differences between those two processes, and some of the transcription factors that we identified as being selected in the HS array can modulate that expression, having an effect on this specific equilibrium. And that could be driving that specific phenotype that we identified, the URA phenotype. Conclusions, we identify a region associated with URA, controlling for case controlling balance, and we were able to identify some potential loci able to modify penetrance for URA. And further research is needed. Thank you.
Thanks very much, Joel. That's great, fantastic. Do you have any questions, Joel? Let's see.
I have one. Joel, in your zoom plot, it looks like the top of the peak is really to the right of the gene. I mean, I know it, it doesn't mean very much, but I'm wondering if you, after having looked at the haplotypes and so on, do you have an explanation for that particular zoom plot?
You mean the distance between the region and the KIT gene?
So the red dots are not over KIT, and
yeah.
the few, the variants that are over KIT are not not associated.
Yeah, we did genotyping for some elements for this specific insertion. The LTR1, and we did a correlation between the most significant marker and the specific insertion, and they are pretty high correlated. So even when they are not totally collocated, there is a high correlation between those.
But those genotypes don't appear in the zoom plot. It was done sort of separately from the only the SNPs are showing here, and you're talking about a structural
insertion.
variant, right? Okay.
Yeah,
okay.
exactly.
Okay.
So it seems that they are pointing specific elements for the insertion.
Mm-hmm.
Joel, there's one more comment in the chat from Valerie. Val, do you want to just ask your question?
Yeah.
Can everyone hear me? Okay.
I don't have voice.
You.
Oh, let me see if I can get my camera to turn on very quickly here. Maybe. Hello. Okay, very cool study. I now work with some ACIs in my laboratory at the Medical College of Wisconsin, and I've noticed that especially in males, there can be issues with the development of different types of the reproductive tract along with renal agenesis. I was hoping you could comment on how some of your results might play into not only the urinary tract, but also the reproductive tract developmental abnormalities that can occur.
Yeah, that's true. So it's a malformation that it seems to affect the same right of the reproductive tract and urinary tract. So there are other tissues that are compromised as well in this malformation.
Great.
This is Jim Scholl. If there's time for another question or comment.
Yeah, quick one, James, please. Thank you.
Yeah, that's very nice. Thank you for your presentation. I just wanted to point out that there's a similar story in the cat. There is a LTR or an endogenous retrovirus story. It's very similar, also an intron 1 of KIT that results in a coat color mutation and deafness. And I think that both of these phenotypes are due to KIT-regulated stem cell migration. So the incomplete penetrance could be easily explained by cells just not getting exactly where they need to be to induce these developmental processes. And so, yeah, I think this is really an interesting story that points to possible roles for these endogenous retroviral elements in lots of complex diseases.
This also has been described for color code in mammals, this specific gene.
Great. Okay, thanks very much for that, Joel. Okay, we're now moving on to our 5-minute flash talks, and we begin with Jiale Zhu from UC San Francisco. Jiale, over to you.
Thanks. And screen. Should I start right now?
That's very good, thanks.
Okay. Do we have a break time, or should I just start right now?
You can start now, thanks.
Okay. Hello, everybody. My name is Jiale, and I'm a postdoc from Sugo's lab in the University of California, San Francisco. Today, I'm just going to present my recent research work about the GWAS for complex behavioral traits using larval zebrafish. So zebrafish as an invertebrate organism, model organism, has been widely studied for many different behaviors, like light dark preference behaviors, signal taxis, condition place preference, and so on and so forth. The release of its reference genome, along with the ease of the molecular genetic studies, have further enabled the investigation of the genetic network underpinning those behaviors. Despite its popularity in post-GWAS studies, zebrafish has not yet been interrogated genome-wide for allelic influence on some complex non-Mendelian traits, leaving a genetic basis under these phenotypic diversity largely unknown. So using a large population of about 6,000 larval zebrafish, here we performed a GWAS for light dark behavior preference related behaviors. The general workflow is to first develop this big population using an EKW, an outbred strain of zebrafish, and then followed by behavior characterization in a well-established light dark paradigm. And in parallel, we isolate the DNA and then send it to our collaborators' lab for genotyping. So once we get all these phenotypic genotypic data, we just fit them in a modified linear mixed model to detect the QTLs associated with our investigated behavioral traits. So to test the behaviors, we carried out weekly experiments, and each week we set up about 20 crosses, that's a pairwise crosses, and then we collected larvae, an equal amount of larvae from each successful cross to make a total of 192. And at 6 dpf, we submit these those 192 individuals in to a arena where only half of the arena is illuminated and the other is not illuminated. So we just recorded their swimming movements in this arena for 8 minutes, and then we recorded like 60 individuals at the same time. So it usually took us about like 2 hours to record all the 192 individuals. And for each individual, we test them in a total of 4 trials, 2 on 6 dpf and the other 2 on 7 dpf. And then those video recordings were digitized in the software, and where we produced a set of raw behavior parameters. And the main parameter we're looking at is called light dark choice index, which is computed by using the the time the fish spent in the light zone and the time the fish spent in the dark zone. And then by definition, it is a value ranges from -1 to 1. Larger value indicates less aversiveness of the fish displayed towards the dark side. So we usually took the mean across the 4 trials as a final value for this parameter for each fish. And the raw data output from the software were then pre-processed for covariate regress out, quantile normalization, and outlier removals to get a phenotypic data set ready for GWAS. After behavior test, we isolated the DNA samples and then sent it to our collaborators' lab, and then where the samples were barcoded and then put together to make a DNA library for sequencing. So samples were genotyped with a low coverage whole genome sequencing in combination with genome imputation. So I'll leave this part of the more details of this part work to our collaborators' presentation, which just coming right after my presentation. So far, about 2,300 individuals have been genotyped. So the genotyping pipeline yielded about more than 10 million SNPs. Following quality control and LD prone, we retained about 2.3 million SNPs for the subsequent GWAS analysis. And these SNPs were distributed all over the 25 chromosomes, of course, with some sort of like non-uniformity. And you can see particularly on the chromosome 4, the large segment of low SNP density, which corresponds to its region on the long arm that has a very a large lack of protein coding genes and an extensive heterochromatin. And for those LD pruned SNPs, we estimate their mean minor allele frequency, which is about 0.16. And we also estimate the LD decay, which decays rapidly within the 1 megabits per SNP distance. So with available phenotype and genotype data, we performed our GWAS, and our initial scan mapped about 17 SNPs for 5 of these 8 behavioral parameters. And then we performed a stepwise regression to combine those correlated SNPs into one independent QTL that is represented by a top SNP. As a result, we were able to find one unique locus for each of those 5 parameters. So with that being done, our current next part of the work will be focused on construct the regional association plot by determining the linkage disequilibrium interval around the top SMP, so that we can get a set of genes that falls into that interval, from where we'll try to investigate about their functionality to pick out the genes with the strongest candidacy for the associated behavioral traits. And one of the techniques we're currently using is called the hybridization chain reaction. And this technique would allow us to quantify the spatial expression of a specific gene all over the brain tissue at the single cell level, so that we can possibly link the differential expression to some sort of SMPs to then to figure out the genes that are most likely to be involved for the traits. And so in conclusion, we have developed a large population, about 6,000 larval zebrafish, using an outbred strain EKW. And then we phenotype them at 6 dpf old and 7 dpf in a light-dark preference behavior paradigm, using a set of 8 behavior parameters. And we also assayed the genotype with more than 10 million SMPs. Our GWAS has already uncovered several independent QTLs for some of these parameters. And we're currently investigating the genes that falls into those QTL loci with the in situ hybridization techniques to quantify their expression level all of the whole brain tissue. So with that being said, I would like to thank the NIH with this funding support, and I'll thank my PI from Guosu from ECSF for her guidance from all the aspects throughout this project, and my research assistant Romelo for his efforts and behavior test. And particularly, I want to thank my co-PI, Dr. Poma from UCSD, along with all the members from his lab for their tremendous efforts in genotyping and their mentorship guiding me through the GWAS analysis. Thank you all. Really enjoyed working with you. Hope we can achieve more in the following couple months. So that's all I want to talk about. Thanks. And I'll be happy to take any questions and comments.
Thanks very much, Jiale. That's great. Any questions for him? I gather the next talk is also about the same system, so you might get questions then as well. Okay. If not, then let's move on to Oksana Polesskaya, who will also be talking, I understand, about the same system from UCSD. Thank you.
Hello, can you see my screen?
Yes.
Okay. I'll just go from this mode. So you can think about this talk as a complementary to the story you just heard from Jiale, and I will talk about how we genotype this zebrafish. So in brief, we develop high-throughput.
Oksana, sorry, could you just go to the presentation view rather than that, because it will be very small otherwise on the screen. If you just, if you go to the bottom right corner, the symbol that looks like a, no, that's the presenter view. You go back to your PowerPoint.
Let me just share a different screen. Share this screen. How about now?
That's perfect.
Okay. All right.
Thanks.
I will be. Yeah. All right. So first, let me describe the population. As Jiale mentioned, we used EKW outbred population from the vendor. It has been maintained by mass breeding for about 12 generations in the Sugio's lab. From this population, we selected some breeders And these breeders generated more than 6,000 larvae for behavioral tests. So I need to mention that we deep sequenced a subset of these breeders, and it's important. It will be important in a minute. For the larva, we developed this method, which I will describe, which I'm describing. And we also deep sequenced a small set of larvae to generate a truth set, which we use for measuring concordance. And this is our sequencing method. We isolate DNA using a high-throughput 96 batches bit-based kit. Then we prepare a library using Riptide protocol. Riptide protocol uses random priming from both ends, and it also can be done in 96 samples per library, very high throughput. Then we sequence the libraries in the batches of 10 libraries per lane, and it derives 0.5x mean coverage. For bioinformatic part, we align it to the genome assembly available from GRC. Then we genotype, then we call genotypes using Stitch software and using these 87 deep sequence breeders as a reference. We only use Stitch for iteration 1, and then we impute genotypes to reference using Beagle. This process, and then we measure accuracy by comparing genotypes with deep sequenced individuals. So this process we call 10 million SNPs with high accuracy. I wanted to point out one caveat of this process. The libraries are prepared in the batches of 96 samples. So within the library, we get a wide range of number of reads, and the individuals with a low number of reads have too few reads to call genotypes confidently. So we end up needing to redo from DNA step, from library preparation step, approximately 5% of samples. So this is kind of caveat of this otherwise very low cost and very high throughput process. As you might remember, we sequenced only a subset of breeders. So the question here was, is this subset, does this subset represent the haplotypes in the population well enough? In other words, can we use just a subset as a reference to confidently call genotypes in all larvae? And this is the experiment which we did to answer this question. We excluded one of the pairs of the breeders completely, and then call genotypes with and without this pair. So here you can see concordance between genotypes when called two different ways. And the individuals in red are the individuals who don't have both parents. So both parents are excluded from genotyping, from the reference set. In blue, only father is present. In green, only mother is present. So as you can see, the excluding specific pair of parents does not really affect the accuracy of genotyping. So this shows that our reference set is sufficient. And finally, I wanted to show you another cool thing, how we use genotyping data to validate and correct pedigree records. It turned out that approximately 10% of samples where pedigree was recorded, it was incorrect. And this has to do with the mechanics of the experiment, how eggs are collected, how larvae are transferred. They just got stuck to the tank and to the net. Since we have genotypes for both parents and offspring, we can correct these genotyping errors. So to do this, we measure Mendelian error rate in each individual. So in this graph, each column is one family. And on y-axis, it is Mendelian error rate. So you can see that in the green box, siblings in each family has very low Mendelian error rate. So this is a good pedigree. But in some families, there are siblings I also need to mention that only problematic families are shown here. There are multiple families which are not problematic. So this is just a subset of problematic families. So some siblings actually have high Mendelian error rate. And this means that they were misassigned to the family. And since we know genotypes, we can assign them to correct family. There is also a set of families where all siblings have a bad Mendelian error rate. And this means that the breeder was misassigned. And again, since we know genotypes, we can correct this error. In conclusion, we developed a low-cost, high-throughput methodology for genotyping zebrafish. It produced that genotyping with high accuracy, and this method enables the first GWAS in outbred zebrafish population, which Jiale presented to you. This work was funded by R122 and it was done in 2 labs, and in Dr. Guo's lab in UCSF and Dr. Palmer's lab in UCSD. Thank you.
Don't see any hands. Oksana, perhaps you can remind me, why would one want to use zebrafish over mice or rats? What are they a particularly good model for?
Yeah, that's probably a good question to Jiale, but I, because he is a zebrafish behavioral specialist. Zebrafish is actually used a lot as a genetic model, but to test specific mutations, because it's kind of very high throughput model. But zebrafish is started to use as a behavioral model. So Jiale, do you want to comment a little bit on it?
Yeah, and I think that to me, like the biggest advantage in zebrafish, especially larval zebrafish, is that we can do the whole brain imaging. So we can just imaging the whole volume of the brain, and like my last slides indicate show that we do the HCR in situ hybridization that allows us to visualize and quantify the expression of a specific gene from not just the one Z plane of the brain, but the whole volume of the brain. So that would help us to further understand it. Let's see that our GWAS pinpoint some candidate genes, and then we can further investigate the functionality at that level. And moreover, you can actually like image that a live fish, what's called using a calcium imaging, that the fish is still alive, and we can image their neuron activities. And then that will also help us to further understand the underpinning physiological network. We're starting from genetic level and then go deeper, so that will make the whole story like more like powerful, and then will well explain why this gene is associated with a certain behavioral traits.
Okay, that's great. Thank you.
Mm-hmm.
Any further questions for the two?
Yeah, hi, this is Sue. I just want to add to what Oksana and Jiale has said about what zebrafish can contribute, or zebrafish GWAS can contribute to the understanding of complex behavior traits. So I think, you know, in addition to what Jiale has said, so zebrafish is really prolific organism. So it's really, you know, great to get lots of animals, and also as Jiale has shown, really high throughput behavior type phenotyping can be done. So yeah, so we can behavior phenotyping 192 animals, you know, in like 2 hours. So so that's really a big advantage. And the secondly, I want to add is that, you know, in larva zebrafish, the genetic contribution to these behavior traits outweighs the environmental contribution, and so making them really attractive to be able to pinpoint the genetic contributions to the behavior, yeah, especially the behavior diversity in the population.
That's great. Thanks very much, Sue, for that. Okay.
Okay, thanks. Hey, I just wanted to thank the organizers for allowing us to really at the last minute sort of group the RGD, or the Rat Genome Database, talks together into kind of a mini symposium. What I would ask is if we could hold questions until after we're done, then we have actually some time for discussion. And I had a couple of questions that I might like to ask the group if we can get some feedback. But in the meantime, I'll let Jennifer start.
Thank you. And I will share my screen. So my name is Jennifer Smith. I am going to change this.
It looks good.
Okay. So I'm a project manager for the biology and curation side of RGD. And I'd like to thank the organizers for giving me the opportunity to give you a very quick overview of RGD as a multi-species resource to explore complex disease. And there we go. So RGD has always been a comparative genomics platform almost from the beginning, because we started with rat, mouse, and human, but more recently we've expanded to include additional species that are used as models for human disease. So in general, this is the front page, and in general on things on the RGD website, such as the tools, default to the rat, because after all, we are the Rat Genome Database. But for researchers looking specifically for data for other species or interested in doing analyses for data in other species, we supply species-specific landing pages, and they're accessed by clicking on a picture of a species that you're interested in on the home page. So to start off, we have human, and each of these landing pages shows general information about the species and its use in research, or in terms of human, the fact that it is what we're all trying to work toward, and then provides links to species-focused data and searches, and links to RGD tools with the species already selected where that's an option. And so we have 10 species. We have 8 species that are fully released to RGD, and 2 more that are coming soon. So we have human, we have mouse, long-tailed chinchilla, domestic dog, 13-lined ground squirrel, bonobo, domestic pig, and then coming soon, we have green monkey and the naked mole rat. So RGD imports gene data from NCBI and Ensemble for all RGD species, and the import of these gene positions, gene model definitions such as exons, introns, and CDSs allow the creation of JBrowse genome browsers for all of our species. So you can see that we have browser instances for all of the species that we have, and for some of them we have browsers for multiple different assemblies. And the data available in each JBrowse instance is specific to the species and the assembly being used. Some have more data types than others, but we do try and get as much data into those browsers as we can so that you can look at the data in its genomic context. So as you would expect, we do have browsers for species like rat and human, which are assembled to the point of chromosomes. So there's rat, there's human, but we also have browsers for species such as squirrel and chinchilla, which only have been assembled to the point of scaffolds. With several thousand scaffolds for each of these, it's harder to browse and find anything, but you can easily find things by searching for instance, a specific gene, by searching for its signal, for its symbol, to go directly to that region of the applicable scaffold. So RGD searches return results for all species where that data is available. So here I've searched for the gene symbol SERPIN-C1, and you can see that I have data listed for all of the species that we have. And if you can also do a gene-specific search and get the genes back for all of the species. So RGD also provides functional annotations for all of the incorporated species. The main gene curation efforts for RGD focus on gene pathway and gene chemical interaction annotations for rat, mouse, and human, gene ontology for rat, and phenotype, that's mammalian phenotype and human phenotype for rat and human respectively. The disease and phenotype, the disease and pathway annotations are assigned across species based on orthology between genes. For the gene ontology, human, mouse, dog, and pig annotations are imported from Uniprot GOA or Ensemble, and for other species, assignments are made based on orthology. And these annotations that are assigned across species form the basis for many of RGD's tools. So you can search by for vocabulary terms and annotations to those terms using either the general search or this ontology and annotation specific search, or for the using the advanced search, Olga, and I'm going to talk about that in just a little bit. And you can browse through any of our ontologies or enter specific terms into this search box to find a specific term across the ontologies. And here I've entered thrombosis, which is the example that I'm going to use for all of my disease-related information that I'm going to talk about. So you can see the search result shows terms from several ontologies. The terms showing in the table are ordered by relevance. If I click on the leaf icon, I can access the ontology browser, which shows the term of interest in this center panel and with all of its sibling terms, and then the more general terms on the left and the more specific child terms on the right. And you can browse up and down the tree by selecting terms in those left and right panels. The list of species under the term above the browser shows the number of annotations to that term and its children for each species. You can click on one of those, or you can click on this A icon beside the term to get to the actual results. At the top, this is the disease ontology report page. At the top of the page, you have your term and some information about the term. In the middle, you have this genome-wide view of the, in this case, genes that are annotated to that term. And this is showing rat. So this is against the rat karyotype. But if I chose human, you can see that human has not only genes, but also variants and cell lines that are also annotated to the term thrombosis. And below that, annotations, you have the annotations with the tabs across the top that show the different species, and then, you know, whatever the data objects are that are annotated. So you can see that for thrombosis, all of the species have genes that are annotated to that term. And in this case, yes, already said that. The symbols link to the object report pages, for example, genes. And this type of data is also available from RGD's disease portals. So RGD's disease portals provide consolidated data across species for 15 different disease categories. If you click on diseases in the menu at the top of just about any RGD page, it will take you to this disease portals landing page, which has recently been updated and streamlined so that it's a little easier to find the disease categories that you're looking for. Here we're going to look at the cardiovascular disease portal. Across the top of the portal are all of the vocabularies for which there is data in the portal, and then the different species with a scoreboard that tells you the number of genes, QTLs and strains annotated to the selected category. In this case, I've selected thrombosis in this embedded ontology browser, and that will let me navigate up and down the tree to find more specific and more general terms that I'm interested in within the context of the portal. The default when you come to the portal is rat, but clicking on another species updates the data in the portal for that species. So if I select human, the portal updates to human data. Below the browser, there's the same type of genome-wide view of the annotated genes against the human karyotype. And below that on the page, there's a list of human genes that have been annotated to thrombosis or any of its child terms, and that can be viewed and downloaded. Below that, there's an embedded instance of our new multi-ontology enrichment tool, or MOET. I'm only going to touch on this very briefly because our next speaker, Dr. Mahima Vedi, will be giving you a much more detailed explanation of the tool. Here, I just want to point out that the portal takes the list of, in this case, human genes that I've just shown and submits them to MOET automatically when the ontology is selected. MOET does the enrichment for any of RGD species and any of the gene-related ontologies. Here I selected the pathway ontology. If I then choose KEBI, the gene list is automatically sent back to the tool for enrichment of the chemical interactions ontology. If mouse is selected as the species, the mammalian phenotype ontology enrichment can be done, and the phenotype enrichment is powered by annotations imported from mouse genome informatics. The annotations are manually curated from the literature and also assigned based on high-throughput phenotype screens, which provides a substantial amount of data, and that increases the statistical power of the analysis. In the tool, in the portal again, gene symbols in the list also link to the corresponding gene page for that species. And the gene page gives easy access to the orthologous genes at RGD, and also to orthologs at the Alliance of Genome Resources, which in addition to mouse, rat, mouse, and human, has data for zebrafish, C. elegans, Drosophila, and yeast. So I'd like to point out that the annotation section in this menu in the left sidebar lists a number of different sources for our ontologies. We have the RGD manual disease ontology, and then imported disease ontologies from ClinVar, the Comparative Toxicogenomics Database, and OMIM, and also for dog and pig, we pull in annotations, disease and phenotype annotations from the online Mendelian Inheritance in Animals database. So each of these links to the section of the page that contains those annotations. And so here you can see that there are multiple annotations to thrombosis and venous thrombosis for the human gene. The ID in the reference column links to the reference report page in RGD, which provides information about all of the data that's been curated out of that article. And I'm not going to talk much about evidence codes, except to say that the ones that are listed here for human other than ISO tell you that there is experimental evidence in that reference cited for the gene in human for the annotation to thrombosis or related terms. If you go to the squirrel page, it also lists RGD manual disease annotations, and these you'll see have the evidence code of ISO that's inferred from sequence orthology. So those are inferred based on the orthology between the human gene and the squirrel gene. And these give the link to the original reference so that you can easily access additional information about the association between the disease and the gene in the original species.
Jennifer, you have about a minute. Sorry.
Okay. I am not going to talk about this other than to say we do pull in annotations from ClinVar, and also we have the clinical variants that we pull in from ClinVar. And I'm going to fly through Olga. This is the advanced search tool. You can look across different species and you can do genomic search by position in multiple assemblies. You can do multiple searches and combine them in a number of different ways. I am not going to talk about that. I'm just going to say if you hit analyze your results, it automatically sends those that list of genes to any of the tools in the RGD toolbox. And I'm not going to talk about the GA tool, but I will mention the variant visualizer. This will also be talked about later in our mini symposium, but the annotation, the genes from the original list were sent to the tool. And if I choose Serpin C1, then I can find the non-synonymous variants, and I can see that the first A variant is actually a non-synonymous variant that's predicted to be benign. If I look at human, I can do searches for the ClinVar variants. Here I've searched for Serpin C1, and I've chosen to only look at pathogenic variants. And I have, this is the list of pathogenic variants for Serpin C1. And if I had talked about it, I would have mentioned that this particular variant was one of the ones that was shown on the Serpin C1 human gene page. And it is a non-synonymous variant, and the top of the page gives information about the position, the nucleotide change, the conservation, the clinical significance that was reported, any conditions that were reported, and then you can see the change in the amino acid and the sequence of the predicted amino acid sequence with the variant amino acid included. And then also we have a variant visualizer for dog. And if I select some of these breeds and look at the dog, I can find a possibly damaging variant in Serpin C1 in a number of different breeds. And with that, I'm going to say thank you and hand it over to Mahima. So I will stop sharing.
Thank you, Jennifer. So Dr. Mahima Vedi is going to be talking about the MOET tool next.
Hi, everyone. Can you see my screen?
Yep, looks good. Thank you.
Thank you. I am Mahima, and I'm going to go over Multi-Ontology Enrichment tool, which is a bioinformatics tool for ontology analysis across multiple species and ontologies. Gene set ontology analysis has become integral for studying complex genomic and transcriptomic data. Most tools for ontology analysis, they focus only on specific genes or proteins or pathways. Therefore, RGD has developed the multi-ontology enrichment tool that generates a list of terms that are statistically overrepresented within the user's gene of interest. MOET provides functionality to perform ontology analysis across multiple ontologies, 5 ontologies for all RGD species. It is accessible and integrated within RGD at analysis and visualization and individual disease portal pages from diseases and disease portals. After clicking on MOET from Analysis and Visualization menu on the RGD front page, select the required species and ontology and enter your gene list as one of the acceptable identifier types, or enter a genomic region of interest. Clicking on Continue shows the results, including downloadable results list and a graph of statistically overrepresented terms with their corresponding p-values, Bonferroni corrections, and odds ratio. The p-value is calculated using the hypergeometric test, and users can view the results in the graph in different ways, including zooming in, lasso select, etc. The option to modify the p-value limit according to the graph to modify the graph accordingly is also available, and the p-value limit is set to 0.05 by default. In this graph, it is changed to 0.01. Users can also compare across species and ontologies by simply clicking on them. For example, this is a graph with humans selected as species and pathway ontology as the ontology selection. Now, also clicking on the buttons that show the number of genes annotated to each term pulls up that list of genes, and explore this gene set sends that sublist back to MOET for analysis. The toolbox icon and the all analysis tools link can be used to share the list of the genes with other RGD tools. For example, use gene annotator to retrieve the functional annotations for genes and their orthologs from all RGD ontologies, and retrieve and find the orthologs in other species and retrieve their genomic positions using GWAS, or find damaging variants using a variant visualizer, as Jennifer mentioned. And as I stated before, diseases and disease portal is another way to reach MOET. Infectious disease portal is selected as a top level term here, and rat as species and disease as ontology category are selected as default. And other ontologies and species can be interchangeably selected by simply clicking on them. Selecting toxoplasmosis shows the associated 25 genes, 1 QTL, and 10 strains as downloadable list. And each of the gene symbols in the gene list is linked to their respective gene report pages, and the same for QTL and strain symbols. And further, users can explore the genomic locations of annotated objects using the genome view over here. Gene set enrichment is provided by MOET, and it is at the bottom of the disease page. Here is the list and the graph of overrepresented terms in rats for toxoplasmosis associated genes in the GO molecular function ontology. Links to other RGD analytical tools and other relevant information is found at the bottom of the disease page, and other ontologies can be interchangeably selected from the top of the disease page from here, or from below the gene set enrichment button. The data sources for MOET include annotation derived from manual curation at RGD and imported and integrated from several other databases, databases. Thus, MOET is an improved tool compared to other ontology analysis tools and is freely accessible, intuitive, and provides functionality to perform ontology analysis across several species and 5 ontologies. Thank you.
Thank you, Mahima. Next up is Shenzhen Wang, and she's going to talk about rat strains at the Rat Genome Database and registration. So, Shenzhen.
So this presentation is about updates on RGD strain. RGD is the authority of race strains and race strain symbols and identifiers. And there are several ways that you can register your strain to RGD. So the first one is the data tab on the drop-down menu. It's in the submission data page, and you can find a strain registration link. And the second place is in the phenotypes and model portals, and the link is under link and resources. From there, you can also submit your strain. And also on every strain page, we have a strain registration link on the top corner. So we provide many places so that you can register your strain. And all these links bring you to the submission form. And here I would like to mention several important components. One is the strain symbol, the background strain, the ILA code, and gene and allele information. And here I would like to point out that ILA code is very important since it is the information to tell user that where the strain is created. And if you have any comments or question about RGD, please click on the small blue icon. And here you will see the thumb up icon. You can either give us a thumb up or send us a message. And we will only require your email to respond to your request. So once you register your strain, you will be able to search them from several places. The first one is from the RGD general search. And this one searches all RGD data. And the second one is the strain search. This one only search strain data. And the third one is the Find Rate Models tool. And this tool is a recent development. So as Jennifer just showed you that the general search give you a data table. And here you under red and strain, you see there are 6 red strains match the search term ADORA2A genes. And this is exactly what you will get if you put the same gene to strain search. You will get a list of the red strain that associated with this gene. And later I will talk about the result of Find Rate models. So what are the RGD ID used? It is used as a component of RRID. RRID stands for Research Resource Identifier. And for example, if you are using the lion hypertension, the AEK strain, the RRID is shown in the RRID portal here, and it's exactly the same as RGD ID, just with an underscore in the ID. And if you are using RRC ID, currently it's still using RGD ID as the ID, but in the future it will change to RRC ID. And so these registered strains are annotated with disease and phenotype annotation. And in this chart, you can see that the congenic is most annotated, and there are about 50% of congenic are annotated with disease or phenotype. And over the past 10 years, we have been seeing a steady increase in the registration of mutant strain in contrast to other strain type. And we are expecting that the annotation of mutant strain will be increasing since there are more publications coming up. So all these strains annotated with disease or phenotype are integrated to our disease portals. For example, in the obesity and metabolic syndrome portal, you will find a table of rat strain models. And for example, here you can find strain associated with obesity and the strain associated with metabolic syndrome. And you can also go to the phenotype and model portal, and in this tool, find model by disease or phenotype. You will be able to search strains that are curated with phenotype or disease annotation from literature. And in this tool, you can either put in a gene or disease or strain symbol, and then you will give you all the strain associated with the disease or phenotype of the keyword you put in. And finally, we would like to thank the researcher who submit their data and also the user who visit us at the RGD website and use our data. So if you have any comment, please send us a message. We will respond to you as soon as possible. Thank you.
Thank you, Shurjan. So I guess I am up next. I'm going to share my screen, hopefully. Let's see. Share. And we'll go to, can you see okay?
Yes, very good.
Okay, great, thank you. So thank you for letting me talk about some of the quantitative phenotype data curation we're doing, and specifically in the HRDP strains, because we've heard a lot about this, this panel in previous talks. So the hybrid rat diversity panel, just as a reminder, is a panel of 96 different strains that were selected to have genetic diversity and are really helpful for genetic studies, especially for complex traits. So the goal of the MCW hybrid rat diversity program, which is an R24 funded to Dr. Dwinell, really is to acquire and rederive the strains that are in the hybrid rat diversity panel, to maintain these and distribute these strains to the scientific community. They're also doing whole genome whole genome sequencing using Illumina short reads. Again, that data will be shared to the scientific community through the public archives. But we're also collaborating with RGD for data deposition. So RGD has developed a landing page, if you will, for the HRDP strains, and this will be centralized and created a centralized resource in order to have phenotypes and genotypic data in this centralized place. So I'm going to talk about the strains and our work annotating their associated phenotypes, and Monica Tutai will be talking about the genetic variation in the HRDP strains next. So for instance, if you click on the WKY in this classic inbred strain from the landing page, you'll be taken to the strain page at RGD for that particular substrain. And you can see the different information that's available, including its origin. And if we know any source data, that's something we're going to have to keep working on to keep updating the source data. But RGD is often, for years, has been doing phenotype curation, mainly for genes, QTLs, and strains. But the main area of curation has been using the mammalian phenotype. And the disadvantage of that is that it is based on abnormal phenotypes. And so some time ago, Mary Shimayama had started this phenominer project to be able to annotate quantitative data from the strains. And in order to do that, you really have to gather some additional information. And so this includes the actual trait, the measurement that is being reported, the method of how that measurement is being made, and any external experimental conditions, including tissue sources or cell sources or other types of experimental conditions. So if we go back to the WKY, you can see that if you on this right-hand side in the summary, you can see the various annotations that have been made. Some of these are disease annotations, such as in the WKY, you could go in and see hypertension. Well, that might be misleading until you go down to the mammalian phenotype and it actually says decreased systemic arterial blood pressure. And then you can actually go down into the quantitative measures so that if you click on systolic blood pressure, it will collate different studies that have measured blood pressure in the WKY. So you can compare and contrast different conditions where you may find increased or decreased blood pressure in that particular strain. So when we went to the hybrid rat diversity panel, we realized that there were several of these strains that had no annotation. So we decided that that was a good goal to do. So we started annotating these 8 strains that didn't have any quantitative data. And the workflow for that is to go to PubMed and start pulling any papers that mention those strains. You have to go through triage to make sure it actually has quantitative data that you can extract from the paper. And then that paper is annotated to bring in the measurements and the values of that particular measurement being done, along with the other terms that go along with that particular study. Now, so far, the group has annotated 128 papers, added almost 3,500 annotations, quantitative annotations for those 8 different strains. And at the same time, in order to be efficient, those same papers are curated for their mammalian phenotypes if there is a comparison strain to identify differences or abnormal phenotypes. And again, those are annotated just as well. So in all, we have curated literature with annotations for disease and phenotype, both qualitative and quantitative, totaling nearly 9,500 annotations for the HRDP strains, along with variants for 48 HRDP strains compared to both RN6 and the new BN rat assembly, which Monica will talk about next. I just have to call out that there are other people working very, very hard on multiple data types and resources for the HRDP, including the hybrid rat diversity program I mentioned, as well as Phenogen. The folks in Prague in Japan who developed the recombinant inbred strains, and the scores of researchers who have developed and who are studying the HRDP strains in their independent research. So with that, I thank you. And I am going to hand off to Monica Tutai.
So my name is Monica Tutai, and I'm research scientist at Rat Genome Database. And today I'm going to present some of the data from analysis of almost half of hybrid rat diversity panel strains, and which were sequenced and provided by Dr. Mindy Dwiner from Medical College of Wisconsin. So I will show my workflow, and with this, I'll talk about the data. And so we received 2 data sets in 2019 and 2020. The quality of the data are pretty good, no problems with that. The only problem we have, it's like up to 19% of the duplication. And as we aim to have at least 30x coverage per strain, that could be problematic. But so as we are using GATK best recommended analysis workflow, there is optimization procedure in a call variance, as this was designed for human population variance analysis, which they usually have the problem with coverage too. So there are steps that are prepared for low coverage. So we'll see how it will work. So next, I will just show a couple of comparisons between 2 different rat genome references, as that was the main interesting part of this analysis, to compare the RN6 genome reference and the new one, which was just released in November 2020. And so we can see that there is a nice improvement in coverage with the new reference. And so nice improvement in the coverage. Still, there are some improper realigned pairs of reads and the chimeric reads, but nothing really serious or important. So in what is the critical step in variant call, we know that it's a base quality calibration. So what does it mean? And usually from sequencing core, we receive the overestimated data. If you will look at this pink bar here, that's where they throw the data in 3 different bars. And what you want to use is a rat training set and adjust the quality score per base based on real data. And the data should look like a normal distribution. So basically all those 48 samples dropped in as quality score like 1 to 9 points. So it is one of the steps which eliminate false negative variance call in future. So the types of variants called between those 2 references, we can see increase in the SNPs and decrease in indels, and more details you will see in next slides. There are also the small fraction of multi-allelic SNPs and complex indels. Usually people don't put much of the interest in this, at least I didn't see many talks about those. And so to be valid variant, currently we are providing data which each variant has to pass all the 6 parameters. So these are stringent criteria, and we know so it's another way of minimizing false positive. And in the future, we will need to adjust and rescue some of false negative results. So here I just compare the studies done over years, and major one which provided a single nucleotide variance data, and these are the 2013, 2015. RGD reanalyzed the 2013 data of the 25 to up to date to the recent rat reference. And currently we can see that we've got 47 rat strains and a nice increase in population of single nucleotide variants. And also some comparison, like, improving of the softwares over the years. And also I will show some annotations, details. So the distribution of single nucleotide variants here, you can see that most of the samples show more than 5 million SNPs called. When we filtered 1 million, basically we are losing. Most of those variants are homozygous calls, but we can see that there's a significant percentage of heterozygous calls. And now this is really interesting because there are inbred rats, and where are those heterozygous calls are coming from? So I try to subset those data and look for the detailed information. And also you can see that there are a few samples, like these 4 samples are the descendant of the rat which was sequenced for the genome reference. There are also recombinant from brown Norway strain, and they are showing lower, generally lower numbers of variants. So if I will look in details for the heterozygous calls, and here now I'm showing comparison between both references, there are no much differences in generally, but there is a one thing here I wanted to point this. These heterozygous call are located, 5% of them, which are dark here, green, are located in chromosome Y and unlocalized and unplaced contigs. So that's interesting. And that's also the reason that we've got the 18 megabase chromosome Y in a new reference compared with the previously assembled chromosome Y 3 megabase. But you could see here, I just, next to the chromosome, I put this distribution of those heterozygous variants, how they, where they are located. So just look at this brown Norway strains and the numbers I'm just showing here in the table. So what is here interesting, that these 2 strains, these are, as I mentioned before, 2 different data sets, 2019 and 2020. So these are the same strain, but sampled twice. And we can see that the other brown Norways, they generally compare between 2 assemblies. We've got a huge drop in the homozygous calls. So basically what we've got left to look closer at a brown Norway heterozygous variant call, which can help us resolve problematic regions still present in a new reference. Also, the number the variant call is much smaller than compared with between the assemblies. So now, looking at the insertion and deletions, you can see generally is a huge drop in a number of indels. So that's interesting, and also something to explore further. We can see that there is also some accumulation in the chromosome Y and unlocalized, but then you could see that these are really heterozygous indels are taking over. The same as in SNPs. It is also more profound with the number of the homozygous calls. So we've got 5 times less in a brown Norway strains. So if we want to look at the functional consequences, so there's another part which we've got this gene model annotations. Yes, so currently we've got NCBI. I just put together all the assemblies available. You can see that we've got a nice increase in a gene over years and the transcript numbers mapped. Currently for NCBI, we've got more of an ensemble. For RN7, there is still available only rapid release, and we can see from our pipeline that each week this number are changing. So it's not still probably complete. And so that's why we use the NCBI annotation for doing the function consequence. This is done by SNP effect tools. Generally, there is no much of the differences. There is a slight difference in the high effect, high impact variants and SNPs, and drop in indels, which probably is also related to what we observed earlier. And we also run polyphen to prediction SNP, that's classified the variants as damaging. And so here's interesting also that we've got practically twice more for RN6 compared with the RN7. So now if we are talking about precision medicine, here's just an example of Olga, which Jennifer mentioned, and this is my favorite tool when you want to play with the ontologies terms. So for example, disease, and I choose here the hypercholesterolemia. You've got the nice list of the genes here, and I want the intersection with coronary artery disease, and look at those just 41 overlapping genes in both diseases. And you can play also with chemicals if you're interested with some kind of the pharmacology studies. So these 41 genes, you can next put in choose some our toolkit tool, and I choose the variant visualizer. And by filtering through different criteria by location in a gene, or the clinical significance, you can select, and this is for human, of course. So as we are starting with the human, and we are looking for the pathogenic variants. So more details you can see per variant. First, you've got numbers per genes. And next, we want to see what we've got in our population of rats. So we are choosing the different assemblies and put this the genes list in it. And the next step is choosing the strains you are interested. So here I'm showing, difference between RN7 and RN6, and right away I can see the nice difference, and that's interesting. I'll look at it, it's real, but if you see something like this, so there are two possibilities. So it's a genome problem or model annotation. So I need to look probably at the PAM files to see how the reads are falling there, if it's a nice alignment, and then the model, the gene model, how it was in the previous assembly, and to see why I don't see those data here. So for future work, that's what we need to finish the whole population of the hybrid rat diversity panel to start building the golden standard variants to have the final score about how real is the variant, and also what we need to label which are not confident for users and researchers to know that this is problematic because different reasons. So the model annotation or the still genome in this area is not finished. And so we uploaded those data on a download site. You can find it through this button here. There is a folder strain specific variants, and the project title is Dwinel MCW Hybrid Rat Diversity Program. And you can find both for RN6 and RN7 data sets, also divided by indel and variants. And here are all the files with index, so we can download them and research by yourself. And you can contact me if you will have any question, or you will need to rename the contigs, or I can provide you all the details. So in summary, I don't know if I've got the time for summary. In any case, what we are trying to generate this population of variants with confidence score. So to assist in finding appropriate disease model for this personal medicine, and the genetic variation in rat strains. So having the good set of variants is essential to build the high resolution associated maps and also to study complex strains. As we know, still I showed you the polyphen, but we need to implement more damaging like sift tools, as we want to see bigger picture, especially between strains, as it's not one gene. Yes, so basically you saw the HRDP portal presented by others, and so thank you.
Hi, I'm Wendy. I'm going to talk a little bit about the updated rat reference and our efforts to curate it. All right, well, the rat is recognized as an important experimental model for many human diseases, such as arthritis, hypertension, and diabetes, and cardiovascular diseases. It's utilized to understand therapeutic efficacy and toxicity of therapeutic compounds prior to human clinical trials. And as mentioned by Gibbs et al., advantages of the rat model are related to its genome. Over 1 billion rat nucleotide bases align orthologously to mouse and human. Rat and human genomes encode a similar number of genes, and almost all human genes known to be associated with disease have orthologs in the rat genome. Currently, there's upwards of 1.6 million publications indexed in PubMed and over $824 million in funding dedicated to disease research with rat models. With all of that research and funding, it's important to have a reliable reference to support it. There have been several updates to the rat assembly since the introduction of Celera's whole genome shotgun assembly based on a female brown Norway rat of generation F14 in 2004. And although improvements have been made with each release, the versions through RN6 have been highly fragmented despite the periodic updates with the addition of new sequence. And as you can see here, in comparison to the well-established mouse genome assembly, the rat assembly quality measures of completeness, such as the N50, which is the minimum length required to cover 50% of the assembled genome sequence, are much lower than in the mouse. The L50, which represents the number of sequences that give you that N50 length, is higher. And additionally, revisions of the rat assembly is noted in the NCBI assembly. It just hasn't been, the updates haven't been as frequent as mouse. So the latest assembly, mammalian rat brown Norway, version 7.2, was released in November of 2020. It was generated from a kidney tissue of a generation F61 male, descendant of the original female brown Norway rat. This assembly, generated by the Darwin Tree of Life project at the Wellcome Sanger Institute, provides major advancements in comparison to the other clone-based rat reference versions. Part of it's through the technology, the integration of PacBio long reads, 10x linked reads, bionanomaps, and the ARIMA Hi-C data. The data were assembled with the VGP project pipeline, version 1.6. And as you can see in the global statistics that are posted on the NCBI assembly page, this new reference 7.2 has some pretty substantial improvements compared to RN6. A few features to note are the counts of scaffolds and contigs have been reduced. The N50 and the L50 measures have improved. And the number of gaps between the scaffolds have been resolved from a count of 440 to 0. Automated annotation of the gene models and alternative transcripts was completed by RefSeq and EMBL. The Rat Genome Database joined the Genome Reference Consortium in August of 2020 to aid in the further refinement of this assembly, initially through the resolution of errors identified by RefSeq, and then long-term through the continued maintenance of the reference. The preliminary milestones for us were to align with the best practices implemented by the GRC for human and mouse reference curation. Currently, myself and other curators and the team have been introduced to and trained on the GRC data formats, common tools such as the NCBI's Genome Workbench and Sanger's Genome Evaluation Browser, and then also the ticket tracking system. And as curators such as myself become proficient with these tools, through the review and the resolution of the issues, then we'll update the protocols for the reference management with the GRC. Issues with the assembly are submitted to the GRC reporting system or directly to us with RGD. The majority of the current issues have been submitted by REST curators or other rat research community members. This is a screenshot of a recent ticket status from the GRC rat landing page. The primary category of issues is classified as clone problems, and this mostly relates to poor quality data, including component misassembly. And then the remaining of the tickets are related to missing sequence. This is gaps within chromosomes, and then order and annotation. This new reference provides us with the opportunity to resolve prior gene model issues related to the lack of underlying sequence, and potentially determine when issues are related to the gene model itself, the genome, or the bioinformatic processes used to generate the assembly. Strain-to-strain issues, assemblies with other data, or transcript issues may also be addressed. It's expected most tickets that can be resolved will rely heavily on data from previous versions of the assembly, unplaced scaffolds, or alternate assembly versions. Additionally, we rely on the contributions of the research community to aid in maintaining a dependable and high quality rat reference. So here's my plug to ask you to please support our efforts and reach out to us with your findings, either directly with us through RGD or through the GRC reporting system. And with that, I'd like to acknowledge the groups and the team members involved in all the talks you've just heard by our group, especially members of the RGD, the GRC, and IROC. And of course, we'd like to recognize the funding from the NHLBI, NIH, and Darwin Tree of Life projects. And with that, I'd like to say thanks for your time and attention. And then Ann Quedick will lead our group's time for questions.